cloud. I'll get the file. All right, well, everybody, welcome to our first brown bag seminar for the 2022 SURF program. This is a program we started last year that honestly, one of the silver linings of the whole COVID pandemic has been is the sort of being able to use these virtual platforms, which I don't love, I'll be honest with you, but for this sort of thing, it actually makes things quite convenient where we can get together from all over the place. So welcome. And I'm recording now. And then, like I said, it will be available online later. Last year's or several of last year's seminars that we did are also been recorded and posted. So if you're interested in these sorts of seminars, you can go back and look at those. You have the various links through our website and whatnot. So it'll be there. Also, BJ told me the other day that several students didn't necessarily understand what it meant to be a brown bag seminar. And it didn't dawn on me that the idea of a brown bag lunch was an old time concept, but I guess it might be now. I might be showing my age, but so I sent out an email today. So I'm just gonna repeat what I said in there, which was brown bag is you bring your lunch in a brown paper bag and you sit around a table and you would eat your lunch out of your brown paper bag and you would discuss various topics. So these sort of brown bag meetings or brown bag seminars is a very common way to sort of interact with your colleagues and whatnot and learn something new. So we decided to do this sort of virtual brown bag over lunch. So if you guys are eating, feel free to continue to eat. And then hopefully at the end of the seminar, you'll have some questions for Dr. Maia. So, okay, without further ado, let me introduce our speaker. Today's speaker is Dr. Anabela Maia, who is an associate professor of biology at Rhode Island College. Her research focuses on fish physiology and biomechanics, especially on how fish respond to anthropogenic pressures. At Rhode Island College, Dr. Maia teaches human and organismal anatomy and physiology courses and supervises multiple research students. She was born in Portugal and completed her initial degrees at the University of Lisbon. She then attended the University of Rhode Island, where she got her PhD in biological sciences. She has held postdoctoral positions at Ghent University in Belgium and Tufts University up in Massachusetts. Prior to joining the faculty at Rhode Island College in 2018, Dr. Maia was an assistant professor at Eastern Illinois University, where she worked on stream restoration and environmental impacts on Midwest fish populations. Dr. Maia has authored 30 peer-reviewed publications in her field. Well, that was last year. Maybe it's been up from that. I don't know, Anabela, but that was from last year. She is active in her profession, participating in many professional societies, such as the Society of Experimental Biology, the Society of Integrative and Comparative Biology, Sigma Xi, and the American Fisheries Society. Dr. Maia is also involved in multiple outreach efforts locally and abroad, including development of science communication materials in Portuguese and science engagement at high schools through data visualization. So without further ado, Dr. Maia, you are ready to go.
Thank you, and thank you so much for coming. And I hope that this presentation interests you. And again, my email is on the screen, also my Twitter handler and the website. You can certainly feel free to check it out and email me if you have any questions. Career, need help deciding what to do after undergrad or grad school. By all means, I want us to, as part of the EBSCOR community, to be a resource for you. So as Jim mentioned, I actually grew up in Portugal. So this is actually a picture of the Tagus estuary, and I'll get to talk to you a little bit about anthropogenic pressures in the Tagus estuary and some of my early work too. But I did go through schooling in Portugal. This is actually the University of Lisbon. These are actually my elementary, middle school and high school. And I did take the journey to Rhode Island to get my PhD. And then after my PhD, I actually returned to Europe, but this time in Belgium, where I was working on seahorses and how they use their tail to grasp onto objects. I also worked at Tufts University. I'll show you a little bit if we have time at the end on the work that I've done there on turbulence. And this is the castle at Eastern Illinois University. Maybe you've heard of Tony Romo. He came from that. Jim Garapolo also graduated from AIU. Maybe you've heard of him. But you know, Rhode Island called and the ocean called, and here I am on the bay, right? Because really, that's where my heart is. That's where my research interests really lie. So I'm currently at Rhode Island College. If some of you might not know where Rhode Island College is, you can see it here up on the top of the bay. So, you know, you have the Narragansett Bay that you'll see multiple times throughout this talk and throughout the summer. And this is just one picture of our library in a sunnier day than today. We do a lot of research in the lab, but also some research in the field. So my students were actually out today fishing. This picture is from earlier in this spring, but they were actually out today trying to catch some silver sides. I haven't seen them, so I don't know if they brought them in or not. But we also try to also have some social times. We just had a lab picnic outdoors at the end of the semester, just to get everyone to know each other. And as we revamp from being online, we need to build those communities and make the most of your labs, right? Your lab spaces are going to be so important to feel that you belong in science. I also think that it's super important to get students to present. And this is actually a picture from last year at our SURF conference with a picture of the lab and some of the students presenting, but also doing some research and just in the field. And I've taken students to conferences like at the Society of Integrative and Comparative Biology, for example. Megan here, I think this one was in Austin, and this one from last year, this past January, was in Phoenix, Arizona. So, you know, not very far from where some of you were traveling earlier this year. But giving you a little more perspective, what are the main anthropogenic pressures that fish encounter? We all know that temperatures are changing. You can see here how the temperature has changed, and all the dark colors means that temperatures are higher than they used to be, right? So it's actually a widespread pattern that actually even is more prevalent in Europe and some other parts of the globe in comparison to the US, but it doesn't mean that it's not a problem here. We also know that this affects circulation patterns. And I think that's important to understand that the circulation patterns have very important impacts in migration of fish. In the temperature is also going to have a cumulative effect. So because of changes in temperature, the circulation patterns can actually be deteriorating. And these circulation patterns are also going to be important to in the dispersal of pollutants. So if you want to think about plastics or other contaminants that we put in the water, they're actually going to be circulating through this circulation patterns. You can also see here that I purposely kept the US, but also Portugal. So for those of you that might not know where Portugal is, it's this little piece of coast. So you're now a little bit more familiar with where I grew up, and because it's such a centered in the ocean country, it was, for me, becoming a marine biologist and addressing all these questions was just something that just came natural to me. So ever since 8th grade, I said I wanted to study sharks and other fish. So and then when I joined college, I said I wanted to be a professor and continue to do research on sharks and fish. And here I am. Sharks are fish, but other fish. So I'll tell you a little bit about physiology of fish and how we are impacting their own environment. I also wanted to change the pointer here to no pointer. I wanted to give you a little bit of why we care and why this is important in terms of the plastic component and the increased contaminants that are going into the bay. This is actually from a project from the Atlantic side of things, so from Europe, but I think it really applies to future efforts that we might have here in the Narragansett Bay. So I'll just play this part here.
One of the biggest environmental challenges that we are facing today is the growing amount of plastic in our ocean. When plastic enters the ocean, it quickly fragments into microplastics and nanoplastics. We can only account for about 1% of the total amount of plastic litter in the ocean. That is, 99% is missing. And this amount of plastic in the ocean actually represents 0.1% of the total global plankton biomass. Atlantico will map the plastic distribution in the Atlantic basin, identify or infer the putative sources, analyze the interaction between plastics and its microbiome, or plastosphere, and quantify the transfer of plastic up the pelagic food web and down toward the deeper ocean depths and the seafloor.
So this is just a brief video so to show you that there is a growing concern about how plastics are actually affecting the biomes and how they're affecting fish. But we need to understand a little bit more how they affect different species and how they affect fish physiology. So those anthropogenic pressures could be of very different sources. I just wanted to change to the pointer. But it's also important to remember that that we actually have the bays and the coastal environments actually offer very important ecological services and marine resources. And I was just checking the chat, but that's Jim, so great. So in terms of marine resources, I'll tell you a little bit more about how the local bays can actually be coastal nursery areas. So that is the place where the small fish and small other organisms seek refuge as they in their early stages of life, so they can have a better chance of surviving predation. It's also important for migratory species to have a place for resting between their migrations, okay, and feed up, fatten up, and also to access the river. So protecting our coastal ecosystems is super important. There's also going to be a component of commercial and recreational fisheries. So I'm sure that right now, if you go out to any pier in Rhode Island, you'll find people like casting their nets, and they're trying to catch the sea bass, but some of them are only catching tautog, and maybe they're too small, or they're looking for flounder. So we also have a lot of shellfish harvesting. Maybe you've seen people walking down a mud flat, looking for quahogs, and there's a lot of farming. So for example, in this picture down here, you can see the location of all the oyster farms that belong to the co-op in the Narragansett Bay. The bay also offers human services. So they also offer like not just recreation, going to the beach, but also like the ability to interact with the environment and to have like a place that you can go walk, say like you go onto the cliff walk, or you just walk outside to like take in some fresh air. And that's super important. You can see here that the Narragansett Bay Estuary Program actually maps like the impacted acceptable and assessed areas in terms of recreational. So for example, areas that you should go bathe in and versus areas that you shouldn't. Okay, and I'm sure that if you've gone down to the beach on Memorial Day, you know that it was pretty busy down south. And you can see here, we have to be also very conscious with the trash that we take to the beach and where it could actually end up, because we don't want it to end up in the stomach of a fish or in the stomach of a whale. So we have a lot of tourism, that people that come to Rhode Island to just to enjoy what we have to offer. So what are the main anthropogenic pressures that animals are faced with in the bay? Again, we talked a little bit about climate change. We also have fisheries and bycatch. So bycatch being that when you catch, we're targeting say flounder, but you catch like any other like species that like sea robins that might be there, that their populations could be, or skates, their populations could be vulnerable. We also have tourism. If tourism is done right, it doesn't have to be anthropogenic pressure, but it could potentially be. Our urban wastewater or runoffs, and also runoffs from the highways, is going to have like a big impact on the quality of our water. And agriculture runoff, not as much here in Rhode Island, but also in terms of in other parts of the country. The industry is also one of the pollutants. So instead of like, we don't want to patch these issues. We want to work with our stakeholders ahead of time, so they can actually find ways that are economically sound for them and that help the environment. So basically, some of the things can be implemented very easily, and it will help us get the like less pollution into the water. But there's other things that we don't think about. For example, boat noise, how the noise actually interacts with the species that will vocalize and communicate with each other, on how we have like boats coming in and they'll have invasive species attached to their hull, and they might actually go into our waters. Trawling can also leave scar marks on the at the bottom of the bay and the ocean. And so there's a lot of different anthropogenic pressures. And I'll talk a little bit more about others, for example, the changes in turbulence and circulation patterns that can impact fish. So this is our first bound bag seminar, and maybe you're trying to find your way a little bit on this larger project. So I figured I would throw here a little bit of what Rhode Island CM stands for and what we've been doing. So basically, we're assessing the biological and ecosystem impacts, and I've been working on that. But we're also predicting the ecosystem response by modeling, and we're also enabling technology that allows us to detect those changes and those problems. At the center is actually what you're part of, the workforce development. We want our surf students to come to be more prepared after their surf experience. So you are in training, right, or workforce training, but there's other initiatives that we can actually have, like the certificate of development, of professional development from the Metcalf Institute, and visualization workshops out of both RIC and the Rhode Island School of Design. There's also other pieces that are going to engage stakeholders, and also the Data Discovery Center has been on this data management part. Okay, so just to give you a picture, the big picture of where this project lies. So once we want to think about strategies that we can actually use the physiology that we know of the fish to actually help predict and also manage species, we can actually come with management tools. And these management tools are going to be ideally species specific, because all species actually work differently, or functional groups. So for example, a flatfish might be closer to another flatfish than like pelagic fishes like tuna or sharks might be grouped in the same category. So we'll also do some food web modeling that allows us to identify the different portions of the food web and allow us to know how they're related and how it's actually transferred, and those bottom up and bottom down effects that you have when you remove or decrease the numbers in one species. Okay, so I don't necessarily work in that, but I can actually give the modelers the information. For example, we've done the respirometry of winter flounder, and we've given the modelers that so they can actually plug that into their models and better predict productivity of the system. Does that make sense? And I didn't say this, but if you have questions, feel free to like raise your hand, chat. This is more of an informal seminar here. So again, this ideally for these measures to work, they need to be stakeholder driven. So we need to actually be in consultation with the fishers and making sure that we're getting listening to what they have to say. They can actually be based on fishery closures or catch limits. Maybe you've actually gone fishing and you know that there's a minimum size for most species, right? So you cannot get like keep a winter flounder that's smaller than X. And that's so that you don't take out the juveniles, and that way you can take more mature fish out, and then you don't deplete the population. So you can see here that we can actually do some modeling of those species, and then we can actually use these models to predict what's the best strategy for fishers and wildlife to actually implement. So I wanted to talk to you a little bit about some of the anthropogenic pressures and their effects on fish physiology that I do in my lab. So we look at the effects of increased temperature. We also look at the effects of microplastics and other pollutants and the impacts of turbulence. So I have a couple of images here. So we know that plastic ingestion by fish is widespread and increasing. So that's an area of concern that we've identified. You can actually do transects down to the beach, and you actually find all the different kinds of plastics, macro, micro, and nanoplastics. And this is actually just a project we did on the exposure to estradiol. Estradiol is a hormone that also results from the breakdown of the contraceptive pill, and is actually used as like added to the the wastewater. So just by regular physiology of women that are taking the contraceptives, and what happens, it can actually impact fish, it can actually change their physiology and their morphology. So I figured I would start a little bit talking about my past experience, because when I was doing this, I was just like you guys. I had just finishing my undergrad, or some of you, some of these things I started even during my undergrad. And we were looking at all the estuaries in Portugal, and we were interested in knowing what the anthropogenic pressures were. So we actually developed a tool that actually could tell you by each estuary which one was more important. So for example, you can see here that the regulation of the banks, the embankments of the estuary, is actually rather important in Rio de Aveiro, in Douro, and in Mondego, but it's not as important in other estuaries, okay? So all of these are different estuaries. You could also see that, for example, aquaculture was actually more important in Tejo and Sado, and not as important in the other estuaries. So basically, has the more important it is, the closer it is to the outside of the circular diagram. And this gives us a tool to identify what are the pressures that actually are affecting each estuary. As you can see, it's a small country, but has a lot of estuaries, and they're all very different. So it gives us a really great place to study. We were able to study the age and growth of flatfish like the Dover sole. We also looked at the effects of temperature on population. So for example, we could actually see that different estuaries along Europe, not just in Portugal, but also in the Northern Sea, actually had a delay in the months that they, well, as we increase latitude, so as you go north, they actually spawn later in the year because it's warmer, okay? So you can see this is December, and this is the spawning period. And you can see that higher latitudes like Netherlands and UK, the Dover sole was actually spawning much later, okay? So when a fish spawns later, they might not have access to as much food as they would otherwise. So then they'll become like, they might not grow as well, and their populations might be more susceptible to overfishing. We also were able to do some really cool research, and we caught the young of the year fish in the estuaries, and then the adults out in the coast. And we could predict and know from the adults where they actually had spawned. We did that with odorless microchemistry, kind of like a fingerprinting. Basically, we sampled the water of each estuary and found the signal in their molecules that were present in each estuary to actually find how much time each fish had spent in there. So basically, you can actually use ICP-MS with laser ablation by looking at the core of the otoliths of the adults, and then compare that with the otoliths of the young of the year fish and see where they came from. So we're actually able to, for each different species, so here you have the, this is one of the soles. This is actually one of the, it's a sea bream. These are two other soles, and you can see the contribution of each estuary. So for example, for this species that's more found in the northern, most of the juveniles or the adult stock came from juveniles out of Douro. You can see that the pie chart, the dark, it's actually where they originated. But for example, the Dicentrax labrax, they actually came mostly of Tejo. That's the white one here in the center of Portugal. While the sole, so this is actually the one that's the Dover sole that's found more in the north. And this is actually the Senegalese sole that actually came from Africa, North Africa, and kept expanding its range because of the increase in temperature. So what you can see is that the Tejo actually produces a lot of soles, but other estuaries like the Mondego is also important in the adult sole stock for the Dover sole. But for the Senegalese sole, it's mostly the Teju and the Sado that are the main nurseries, and they even like export individuals all the way to the south of Portugal. We also did some other work on metals, heavy metal contamination, because soles actually live in the bottom, so they eat a lot of the polychaetes that are found under the sediments. So they're like really good at bioaccumulating. So it's not probably the best fish to eat if you have concerns about the pollution of the sediments. So the last one I wanted to tell you about from our work in Portugal is actually related to gastric evacuation. So think about the time that it takes for food to actually go through your gut and the effect of temperature. We also look at salinity, but how that differs in the two species, the one that came from Africa and the one that's more of a northern sole, the Dover sole and the Senegalese sole. And what you have here is actually the temperatures that they were at, okay? And what we did is that we would euthanize fish at different times after their meal, and we'd figure out where the food was in their guts. So you can actually see that the curve is much steeper when you have warmer temperatures, as you'd expect, like for the sole Senegalese. So this species can actually digest food really well at the warmer temperatures because that's what it prefers. But you might notice that in this case, they were actually having a hard time. So the Dover sole is actually challenged at that higher temperature. And that higher temperature is becoming prevalent in the estuaries in Portugal. So this is becoming a problem, a physiological problem for the fish. They're not able to digest and get as much nutrients out of their food, so they're actually going to have delayed growth and issues. So you can see here that these curves are they're actually at least progressing, and even though it's actually a little slower as you'd expect at lower temperatures. And recently, we've actually been able to get some of this data in GeoPortal. So for those of you that are interested in GIS, we've been able to get all this information into GeoPortal and even get the different diet information of the fish, getting the abundance, getting their growth rates, so that even fishermen, if they want to know, they can actually go to the portal and get that information. So widely available. Kind of nice to go over and see, like, it's been a long time since this work has been done, but it's kind of interesting to see it again, like in available to the public. And that actually brings me to something that we've actually been working on. That's our simple charts Rhode Island. So, if you actually just want to visualize data from the Bay, the Data Discovery Center is amazing, but we also have a tool called Simple Charts RI. I'll show that in the end if I have time. That actually has you can plot, this is open source, web-based, so you don't have to worry about it, but you can plot the capture per unit effort. So how abundant the fish is in the fishery from the historical data all the way to 1959. So it's very interesting that there's all these parallels between what I was doing in Portugal, where we're able to get the historical data and see how the fish are changing, and what do we know about Narragansett Bay and the plethora of historical data we have available to predict and to know what's going on in the Bay. So now I wanted to talk to you a little bit about the effect of temperature on muscle mechanics and also respirometry. So some of the projects that we've been doing under Rhode Island CM in my lab. So what we did was that we actually raised the fish at different temperatures. We actually have now more than n equals 4. We have n8. And then we tested them at different temperatures. And we were able, this was the work of Megan and Jesse, and we've been continuing that since then. We're able to measure their oxygen consumption at different temperatures, but also their muscle activity. So fish have two types of muscle, white and red muscle. Most of fish have that type. And you got to think that the red muscle is actually aerobic, needs a lot of oxygen, and it's what they use for sustained swimming. And the white mussel is anaerobic, and it's what you use for sprinting. So if fish wants to escape, they use mostly white mussel. If they're swimming for migration, they're using mostly red mussel. So we were interested in knowing how fish species were affected by the rising temperature, mostly and in terms of how their muscle mechanics for the red and the white mussel would change and how their oxygen would consumption would change. So what Megan and Jess did was that they actually were looking at the magnitude of the mussels. So I'll walk you through this, but pink and red means red mussel, and gray and white means white mussel, okay? And here you have on the left the winter flounder, the center the scaup, and on the right the black sea bass. And we also looked at two positions along the body. So we looked closer to the tail and also closer to the center. And what you can see is that winter flounder, most fish, as you increase temperature, red mussel will respond by increasing in magnitude. But in the flatfish, they're not able to do so. You can see here the trend line for the red caudal mussel is actually decreasing. So it's too warm for them. They're not able to use that mussel correctly. Now, the white mussel is not as dependent on oxygen, so maybe this is related to oxygen delivery to their muscles, okay? Maybe it's related to the enzymes that actually help with the contraction of the muscle. But winter flounders seem to be in trouble. You might actually guess that what fish is the least in trouble from these three that you see here, just by looking at the graphs? Any guesses? You can unmute and say it.
The bass.
The bass, exactly. So the bass, like they're happy. Like, I mean, sure, there's a little decrease on the white mussel, but all of them are increasing. So these bass, like they don't really mind the temperature. They've actually been increasing their distribution and coming more to the bay from the south. So it's not that surprising. But we're seeing a problem with the red mussels. That's what the fish rely on for swimming around all the time. In both the scaup and the winter flounder, and this actually could explain why this some of these species are actually decreasing in the bay as the temperatures increase. Okay, so we know that there's very different strategies in terms of the power that's available for their muscle. We also look at duration, so how long the muscle is on, and we know that they're actually like the bass is actually increasing power temperature, but the scaup and the winter flounder are actually decreasing power with temperature. So we really already know that these species are very different in terms of their physiological adaptations to temperatures. But we can also look at their metabolic rate. So we can actually look at the change in oxygen consumption from a baseline of 20 Celsius to 22 and 24. And I have some other graphs that actually look at the black sea bass and scaup separately in more detail. Basically, you just put the fish in a chamber, you get an oxygen probe that's actually going to be measuring the oxygen every second, and you can see how much oxygen they're consuming. And remember, if you remember, the black sea bass actually had a lot of the muscles were increasing in their power, right? So this is what I would expect. This is the normal behavior where that as the temperature increases, the oxygen consumption increases, okay? But sometimes if you're really stressed, you also feel that you start like breathing heavier and heavier and heavier. And that can actually be a stress mechanism. So, you know, your oxygen consumption is actually a proxy for how stressed you are. So what you can see here is that the scaup and the flounders actually have different strategies. The scaup is actually increasing oxygen a lot and getting really out of breath. They're not able to, they're not that comfortable. But the winter flounders are actually sort of decreasing their oxygen consumption as they are increasing temperature. And that's a strategy known for some organisms that actually depress their metabolic rate when they're uncomfortable, so they can actually store energy. Okay, so we're actually seeing like different strategies that are found in the scaup and the flounders in the bay. We have actually been able to increase this. This is the work of my grad student, Isabel Cote, that you see there on the top. by looking at the two different types of metabolic rate. So you can actually look at the metabolic rate of a fish at rest, or you can actually look at the maximum metabolic rate. So this is like how you chase a fish, and then like when they're tired, how much oxygen they're actually consuming, okay? So what you can see is that there's a little increase from 18 from the basal to the maximum. But then and for the scup, like there's from the 20 and 22, they're still like they're able to increase their maximum metabolic rate. But at the 24, when they're already really stressed, their oxygen consumption, the basal oxygen consumption is so high, they don't have a lot to go by. And then they're actually like, that's another reason that they might not have enough energy to grow, because they're maxed out at their oxygen consumption. We actually seen a very different pattern in bass. So bass, like, oh wait, I have a lot of what we call aerobic scope. Aerobic scope is the difference between that maximum metabolic rate and the basal metabolic rate. So they have space, and we actually don't have data for 18, so this is not zero, it's just not there. But they have space to keep increasing and keep getting oxygen to those muscles. So Black Sea bass are actually much better at performing at these higher temperatures, and they can actually, they're actually more comfortable at higher temperatures than the other species. That makes sense? Okay. So we've also been collaborating part of a STAC grant with Dr. Colleen Suckling at University of Rhode Island. We were looking at the effects on temperature on planktivorous fish. So we did a similar experiment, but we were actually putting the silver sides, alewives, and menhaden in little like tubes to test their oxygen consumption. So this was done during the pandemic, so it was a little crazy, but it was also pretty neat to keep research going. So here there's like those little silver sides. Now our chamber is much smaller, because ideally your chamber should be about 10 times larger than the fish in mass, so that they don't exhaust all the oxygen. So we did respirometry, and I'm mostly showing you the alewife and silver sides data, but we did respirometry. We also did RNA DNA ratio. And does anyone know what RNA and DNA ratio help you with? Do you know what you can actually figure out from the RNA DNA ratio? Do you know if a fatter fish will have a higher RNA DNA ratio? I'll explain to you, but basically DNA is going to be constant in all your cells, because DNA is actually pretty much the same. But RNA is going to measure protein synthesis, right? And whenever you have more food and you can grow more, and I said fat as in like you have excess of energy, you can actually produce more proteins. So the RNA DNA ratio gives you an idea of the condition of the fish. So the higher RNA DNA means that the fish is healthier than a fish that has a more depleted RNA DNA ratio. We're also looking at agent growth studies. So you can actually see here, these are the otoliths at ambient and +2 Celsius of increase in temperature. And we can actually measure this is ongoing research, so we don't have all the numbers yet, but basically you can measure the growth of the otoliths from the center of the position and figure out how much the fish is growing. We can also look at the respirometry, just like I showed you before for the other fish. We can look at the oxygen consumption over time, and we can fit a regression to that, aligned to that data. And we actually use the slope of that data to actually figure out how much oxygen was consumed and multiply that by the chamber to get the volume, okay? And we're actually feeding this information into modeling efforts with DEM. So they're actually predicting how this planktivorous fish will be affected by temperature. So I'll show you a little bit of that, of our work from that. So this is actually all the fish, all the trials that we actually collected. So baseline is just before we actually start any experiment. So straight out of the field. Ambient is the temperature that the bay is at, and IPCC is actually an increase of the ambient temperature by 2 degrees. So in this case here, the alewife, when we got them out of the field, they actually had a little higher oxygen consumption. This is not surprising because we actually caught them during the warmer months, okay? And they probably could be a little stressed even though we held them for a week after they were caught. But then what we see is that in comparison with the ambient and the warmer temperature that we raised them, they were not significantly different. So they actually were similar values of oxygen consumption at these two temperatures. But we actually had a regression signal when we actually looked at the mass of the fish. So oxygen metabolic rates normally by the mass of the fish. So what's happening here is that the mass is actually masking the effect of what's actually happening. What we do see is that at cooler temperatures, okay, as the fish increases, the metabolic rate does not increase as fast as at higher temperatures. So we know that this is actually affecting the fish, but it's affecting the fish more depending on their size. So there's a size and temperature interaction factor here. Okay, so we do know that the oxygen consumption is changing, but maybe the fish, the larger fish, are actually increasing, getting more lower oxygen consumption. So they're actually so here this is lower. So they're actually like getting their oxygen consumption more depressed than the smaller fish. They're actually able to well, you know, they're actually having a higher oxygen consumption. Now, this higher oxygen consumption does not necessarily mean that the fish is at a better condition, and that's why we want to look at the RNA DNA, because remember, it could be a sign of stress. It could be a sign that they're actually spending so much energy just breathing and maintaining their body, they don't have time to grow. So we looked at the RNA DNA, and this is actually for alewife and for silversides. And this is actually the RNA DNA ratio for baseline. And then we have the condition index at ambient and the condition index at warmer temperatures. And we're still like not finding a specific, a very strong signal, mostly because of the range in sizes. So we want to go back and look at the different sizes. But what you can see here is that the distribution of RNA DNA at the higher temperatures is lower. So in general, the fish at higher temperatures are in poorer condition. Okay, and that's true for both species. I'm gonna go ahead and change my pointer. So I don't want to like go too much on this, so I want to talk a little bit more about and give you some time at the end to ask questions. A little about some of the projects, outreach projects that we've been doing. So we've also been doing some science. I told you in Portuguese, right? So some books about the talking to Portuguese. It's so important. Talking to scientists, Portuguese scientists, so important for people to have a connection some way or another. So I've also did a really cool thing. I talked to kids from K through 7th grade in Germany, and we did an experiment. Like we just built a fish out of a bottle, and we actually looked at how the fish actually behaved in different types of water, and if we added like some weight to the bottle, etc. So that was really neat to be able to do that over Zoom again. And we just wrapped up our Ready, Set, Visualize, a workshop with high school teachers so they can actually use simple charts in their classroom. And if you are a little curious about simple charts, like this is mostly the work of Sally Ann, Muda, and Sean that you see here, but the link is up on top. It's very easy, simplechartsrhode-island.com, and you choose your data and you select the chart type and you create a chart. And you can see that there's simple data sets and there are also complex data sets. And they're going, some of that, the top ones will be related to the Narragansett Bay. So you can play with it. And maybe if you ever, you can input your own data. So if you want an easy way to make graphs during the surf, you can certainly use simple charts too. All right, with that, I just wanted to give you a couple of take-home messages. So again, health of fish populations is really impacted by the rapid changing environments. We need to be sure that we actually understand at the physiological level how that is affecting growth, reproduction, feeding, and the general health of the fish. We want to make sure that fisheries are sustainable, because that will also help us keeping diversity and enabling a stronger blue economy. And we also want to make sure that this goes up to the management strategies, and we get the information from the stakeholders to close the loop. Just wanted to show you some of the acknowledgements, all the people and funding sources that made this project possible. And with that, I'll take any questions you have.
All right, thanks, Dr. Maia. Hope everybody enjoyed that. And if you could turn your cameras back on now, that would be wonderful, so Dr. Maia can see your faces if possible. And then you can raise your hand as Dr. Margaret has just done there in the top there. And so, and I'll let you guys ask some questions, and I might have some too if there's time, but I'll wait.
Go for it.
All right. Oh, we're going to echo. I'm going to go out in the hall. Sorry, we've got a lot of computers going at once. So thank you for that. I was glad that I did not chime in when you asked what we thought about the RNA DNA ratios. I was a little surprised. See, my instinct was that maybe you'd be producing more RNA or detect more RNA because they're responding to stress and making proteins to compensate for that. Does that happen a little bit? And I get that it might not happen in a way that you could detect it at this macro level, but I'm wondering if you could just maybe elaborate on that for us.
That's a great point. I mean, the RNA could be upregulated. You could see more RNA if they're upregulating stress proteins, right? That is true. But overall, there's actually been a lot of studies that actually shown that RNA increases with growth and increases with high condition. So just because general processes actually will be down regulated during the stress response. And maybe what you'd see is that an acute RNA-DNA ratio, if you were measuring at the cell level, it would actually see that, or if you were measuring at the gills or something like that. But we're actually doing pretty much whole body. We take the head and take the guts and just do mostly muscle, right? So we're actually getting more of an idea of what's happening in terms of growth, adding of protein. So yes, at specific tissues, that could be definitely true.
Cool, thank you very much.
These are tiny fish. That's another thing. Like, I mean, the whole thing is this, and then we want to quantify RNA and DNA, and just like we have not much left. Any other questions? Questions, curiosity, anything that,
Hi, yeah, I have a question about, we're talking about how the bass with an increase in temperature, they would consume more oxygen and increase power and everything, but not the flounder. Is that something that you think over time, fish like the flounder would be able to adapt to? Or do you think that with the increase in temperature that's happening and it's going to happen in the future, do you think that that's going to kind of wipe out the species that are unable to have more power in these increased temperatures?
That's a great question, and we're trying to address that in the lab right now. We're actually raising silver size at one temperature and then testing them, and then revert at a different temperature, and then reversing that, and then testing them again, because we want to separate the two factors: the adaptation factor, right? How you can actually build your the amount of enzymes you have that optimize it to a certain temperature, versus what you'd actually have if you are going to the immediate stress response to that temperature. Now, because we sort of for the second experiment, when we actually look at the RNA DNA and also the oxygen consumption on the silver size and the alewives, we actually maintained the fish at a long time at that temperature, right? So we actually were able to see that those patterns would actually hold. in terms of the flounder specifically, they're not really adapting as fast as we would want them to. I mean, they might just be maxed out at the genotypic level. So maybe if we had enough reproduction over time, we would actually have morphs that would be more that would take over. But sometimes evolution of a species has been around with the genetic makeup for a while, adapted to an environment, takes longer than other species just occupying their range. So you saw that on the two souls in Portugal, and how like one soul has been occupying the range of distribution because it's more tolerant to high temperatures, spawns more, grows more, etc. So yes, those are great questions. And for the flounder, I would say that they're not able to do that yet, but maybe the scud can over time overcome that. Did I answer your question, Cassidy?
Yes, thank you so much. It's very interesting.
Yes, unmute for your students.
I'm going to be going in the hall as well. So I know that various fish are having various reactions to the increase in temperature. And I actually work for DEM, and I've seen that some of the populations of fish have adapted to this by shifting their population northward along the Atlantic coast. So like black sea bass is one that they saw this with, where the largest biomass of the population has begun to shift up the coastline. I was wondering if you had any thoughts on if flounder would be able to adapt to this, as well as what the ramifications for this are going to be on our habitats, because like black sea bass are really voracious predators and have started to attack the Rhode Island shellfish population. So I think we're going to see a lot more of that, and I was wondering what your thoughts are.
Those are great questions, and I'm glad that you're actually seeing that in the in real life happening, because that's what we also like the information we're getting, but we're not getting as much because we're focusing on Narragansett Bay. We're not focusing as much on the distribution. The key there, you just said that black sea bass are actually very voracious, and they can actually change the whole ecosystem, and we see that a lot with species. But the problem with the flatfish is that their ranges are pretty small. So their biggest dispersal is actually from the juvenile phase. So they'll go out to the coast and they can actually migrate a little bit, mostly through currents. But they're not as able to, if the asteroids are very far, because they rely on them for the nursery areas, they're not as well equipped as a black sea bass to just increase their range up north. So we're seeing, for example, with skates, there's more plasticity and they're actually changing along the coast. But unfortunately, we're not seeing that much with the flounders. In terms of the landscape, I mean, yes, it's going to be a top-down control on our benthic communities, and it can actually have other impacts on even just food availability for native species.
All right. Thanks, Anabela. This was great. We're right at 12 o'clock or 1 o'clock rather. So I think we'll end this here. But I suppose if folks still had questions or wanted to chat, maybe you hang around for a minute or two afterwards just to see if people wanted to. But I want to thank everybody for joining us. If we could, oh good, I can see myself on the screen on the lab wall there. Sorry. If we can give a little round of applause for Dr. Maia, and thank you for coming. And I appreciate everybody being here, and I'll see you guys later.
Thank you.
All right, I'm going to end the recording now.
I actually have a question.